This protocol describes how to properly dissect flower buds, flowers, and young Siliques, and the subsequent hole-mounting clearing for diverse staining and observation techniques.
This method can help answer key questions in the sexual plant reproduction field, such as sterile mutant analysis or any environmentally and experimentally induced failure in flower development and function. The main advantage of this technique is to allow for easy and quick screening of many developmental stages simultaneously. Visual demonstration of this method is critical as flower organ dissection steps are difficult to learn because they require prior knowledge of flower anatomy and dissection scales.
To begin, use small scissors to remove flowers from synchronized plants, and immediately place them into individual microcentrifuge tubes containing an appropriate fixative. Next, remove the fixative and add enough 70% ethanol to completely cover the samples. Return the samples to 4 degrees Celsius for at least 24 hours. Then, fill a watchmaker's glass with freshly made 70% ethanol and place it into a small petri dish for support. Then, place the inflorescence into the watchmaker's glass and ensure it is totally covered by the ethanol. Using forceps and a syringe equipped with a needle, dissect the flower under the stereo microscope by tearing apart the sepals, petals, and stamen. Then, place the watchmaker's glass aside.